Well, thanks for being here. We're going to get started on the first part with the MGSurvE framework. As I was mentioning, all the materials are available on my GitHub repository. It was shared with the flyer for the webinar, but anyways, we will be sharing it if anyone wants access. Well, the access is public, but if anyone wants the link. There's a couple of ways to get the materials running, as I was mentioning. One of them is installing Docker in your computer. Once Docker is installed, you just need to run this command on your terminal to download all the materials. Everything should be downloaded automatically. And then this second command should launch the materials with all the packages installed. Everything that should be needed to run materials should be run with that one. And after that, we just click this link, and it should launch a screen on our Internet Explorer or Chrome or Firefox, and that should run. We can run the materials right there. Just one quick note. If we want to save changes on the notebooks themselves, like, for example, if we're taking notes or making changes on the notebooks that we want to save for future sessions, we need to create a volume and run it and attach the volume through this command. Otherwise, the changes we make on the notebooks will be volatile. So each time we run the notebook, it will be a fresh start. With those notes out of the way, we can get started. Oh, and if at any point there's any questions or any comments, feel free to jump in. It's probably easier and better for everyone than waiting till the end. So yeah, this is our new software for, this is the MGSurvE Python package. The main goal, I'm going to borrow a couple of my slides for ASTMH. I'm going to be giving a talk at ASTMH if anyone wants to come. So mainly going through why we are building this software. So MGSurvE, its main objective is to be able to optimize the locations of mosquito traps in heterogeneous landscapes. And to answer, for example, the question, given a set of traps, of N traps, and that mosquitoes are flying in a certain pattern in a landscape, where should we place the traps to catch them, to detect transgenes or resistant alleles, insecticide-resistant traits as quickly as possible? So we can imagine, for example, in this scenario, that all of the triangles, circles, and squares are locations to which mosquitoes are flying, and the lines, the connections, so the probability of flying from one point to the next, and the traps are the X's and the circles. We want to know where we should locate these traps so that we can detect these transgenes as quickly as possible, and that's what this software aims to do. The way it works, it's it's fairly simple in general. That's one of the things I like about this one. It takes if mosquitoes, one of the assumptions it makes is that mosquitoes would be flying in a Markovian manner. So it only matters their current state to know the probability of flying to the next site. And what it does, it assumes that the traps are absorbing states so that mosquitoes who fly into traps aren't able to escape. And it optimizes the Markov fundamental matrix that minimizes the time, that detects the time it takes for mosquitoes to jump into those states. And then we feed that into an optimization cycle so that we can place these traps on the landscape. And we do this, as I was mentioning, mainly to be able to detect these transients in either gene drives for trials, or insecticide resistance, or general surveillance could work. Like, for example, if we are planning a field trial of a gene drive in any heterogeneous landscape, we can optimize the landscape so that we can detect the transients spread as uniformly as possible. So yeah, with that out of the way, we're gonna be going through these four parts of the webinar. The first part is just the fundamentals, just a quick rundown on how the software works. And the third, well, the other three parts are the main parts of the pipeline of how MGSurvE works: the setting up the landscape, the optimization, and the analysis. So regarding well, I don't know if there's any questions or comments so far. Otherwise, we can get started with some code. Well, let's get started. So in general, the way MGSurvE works is first we need to set up a landscape. In this example, we'll be setting up as a random landscape that has this general shape. We will be setting up a donut shape environment, which is usually good for testing these optimization cycles. And we will be laying down 2 traps with different attractiveness kernels. These kernels define how likely a mosquito is to fly into the trap given a certain distance. So for example, if a mosquito was standing on the trap, in this case, the green one, the mosquito would have a probability of 0.5 of falling into the trap and 0.5 of just flying away. And the sites in this case have all the resources that a mosquito would need to survive. So it's homogeneous in that regard. It doesn't matter to which point it flies. And this is the way we usually set it up. In this case, as I was mentioning, it's a random setup of points. So it has its X, Y coordinates and the point type, which is the same for everyone. We set up a mosquito movement kernel. I don't think I plotted it in this one, but I know there's a plot of that in a slide down the road. But in this case, it's this kernel defines the probability of a mosquito flying from a given point to another one, provided a distance between them. In this case, it's an exponentially decaying shape. It's similar to this one, but it's contingent on a 0.25 probability of flying or staying in the site. And we set up some traps. Right now, it doesn't matter where we set them up because we're going to be optimizing it. And their kernels, as I was mentioning it, we have these 2 attractiveness kernels, and we're going to lay down 2 traps with 1 kernel and 2 traps with another one. Once everything's set up, we can generate our landscape object like this. We set up the points, the kernels, the traps, and the traps kernels. And we're on our optimization cycle. I'm not going to run it again because it takes a couple of seconds. But once it's launched, we can see this. The most important column in this whole thing is the mean. And what that column is doing is minimizing the time it would take for the average time it would take for a mosquito to fall into a trap, provided that it started anywhere in the landscape. So in general, we want that number to be as low as possible. That would maximize the efficiency of our traps. And we can see that it's decreasing as the optimization goes by. And in this case, I just ran it for 100 generations, which is really low. So this is likely not the optimum, but we can see that it's not bad. That we would expect it to put, well, in a uniform landscape, to put the traps in a uniform way. And this is what we get in this case. If we run it for more generations, it would probably be better, but that takes a bit more time. So yeah, in general, this would be these colors correspond to the kernels here, the 0.75, the 0.5 are these 2. And the shaded regions are the probabilities of, well, the probabilities for mosquitoes to jump into the traps. They are just for visual reference. The circles don't really matter that much, but it can be disabled, but it's a good reference just to know which traps are which. And yeah, that's it for the quick start. I know I'm going super quick on this one, but we'll be going through some of these parts in other examples. So this is just like a quick overview of how everything works. And yeah, I don't know if there are any questions on this one. So I'm going to give a bit more explanation on the three main stages of the pipelines for any MGSurvE optimization task. The first one is setting up the landscape. So one of the things we wanted to be, well, we focused on this development was to make the landscape as flexible as possible. In the previous example, I showed the optimization of a flat landscape of a Euclidean geometry. So the coordinates were only X and Y. But MGSurvE can handle easily longitude and latitude coordinates through some other packages, and it can use either Vincenti or Habersign, or whatever distance functions we need. And it can also handle multiple resources. So in the previous example, I showed a landscape that had all the resources distributed homogeneously amongst the point types. But if we imagine an example in which, for example, the circles were aquatic habitats and the triangles were bloodhounds, we can incorporate the probability of mosquitoes being alternating between the aquatic habitat and the bloodhound more probably than jumping from aquatic habitat to aquatic habitat. So for example, if all the points were equally as probable, we would have this shape on the homogeneous site type. But if we assign them a probability jump between them, we can make the transitions between, for example, let's say 1 and 6 more probable than 1 and 4 accordingly. And the optimization cycle would take that into account. At the moment, it's placing all the traps, which is one of the features we wanted to include in this landscape. And there's other things that are, we can also accommodate points being more attractive than others. There's a vector that could make some points more attractive to mosquitoes than others if we have some evidence for that. But we won't be going through that one in these demos. The next step is to, well, once we set up our landscape with all the probabilities and all the movement, well, actually, before that, another option is that right now, for example, in the examples I've been showing, all of the movement matrices between the points in the landscape have been calculated within MGSurvE. But for example, if we have evidence from the field or from many other sources that mosquitoes are flying in a different pattern than the one that MGSurvE would provide, that can be incorporated, given a matrix in which we can calculate the probability of a mosquito flying from one point to the other one. We can feed it directly to MGSurvE, and that would work. We will show an example of that later on with a matrix that Thomas Leon generated for the last couple of years ago. Once we set up these landscapes, we can go on to do the optimization. In its initial form, MGSurvE uses genetic algorithms to do the optimization. We will show in a bit how this is done. We won't be going too deep into that because it's a lot. But in general, MGSurvE can optimize both continuous and discrete landscapes. So for example, in a continuous landscape, traps could be located anywhere in the coordinate set within a bounding box. And in a discrete landscape, the traps would only be placed inside that are defined. So for example, if we were doing the discrete optimization, we would be just placing traps either in 1, 2, 3, 4, 5, or 6 in this one, not anywhere in between. The continuous one is better for urban landscapes that are smaller and we could place traps anywhere. The discrete one is usually better for villages and larger landscapes in which we just have a predefined set of locations where we can set them. But yeah, that's the main thing with the optimization cycle in MGSurvE. It uses an external framework to do this. It's a really good one that it's fairly optimized. And other fitness functions can be used if we want to optimize for the maximum, to minimize the maximum time to detection instead of the average, and things like that. It can be extended. And yeah, this is usually the step that takes the longest. It's just the nature of optimization. And after that, we usually do some analysis and verification. We can export the landscapes, the matrices, all the objects we need to rerun the analysis to disk. We provide all the functions to do that. We can do the plots of the traps. All of these plots are have been done with the package itself and some libraries. They can be done for both Euclidean and realistic landscapes in sphere, in the planet. And we can check the performance and rerun optimization if we need to. But in general, we will be doing these 3 steps every time we run an optimization task. And that's it. We're gonna start with the landscape. Just go through some really quick examples of how to set up landscapes. In this one, for example, let's say this one will show a bit of how the framework works in terms of setting up the point sets. This first one is fairly simple. We will be laying down 4 points. 4 points, yeah. And with this point, we will be running, we won't be running the optimization for this one, but let's say we were setting these 4 points. And 2 traps. The way we do this in Python is just we need to set up a data frame, which is almost the same as we do in R. We can just generate it in different ways, but it really doesn't matter how it's done. As long as we end up with a data frame that has either X, Y, and type, or long, lat, and type, we can set it up. With this, we also set up our traps. If we're going to optimize the traps, the location doesn't really matter. But for this example, I set them up manually so that we can check the time to capture. And that's pretty much it in these cases. We just set up the kernels and we launch the landscape, and we can see that it's being plotted here, the 4 points with our 2 traps. This matrix is usually just for reference, but we don't plot it almost. We usually don't use it. We just use it for these examples because it's showing how the traps are absorbing and how the migration matrix is modified. But here we have the time to capture, which in this case would be 11.08. But let's say on the mosquito movement kernel that I didn't show in the previous one. So for example, mosquitoes have a 0.5 probability of remaining in the same place. And then the probability decays as the distance increases. And let's say we wanted to update the traps. We will be adding another trap, but with a smaller kernel, and shifting the traps in place. We do this for a couple of commands, and we can see that the time to capture is being reduced. This location of the traps is way better than the one we previously showed. And this one is right on top of the site, so we would expect the time to drop. And it is what is happening. We're getting a better time. In this case, I'm doing it manually, but that's why we have the optimization step. The optimization algorithm does that for us. So that's the nice thing of automating this, of having a metric that we can automate. And that's it for this one. But let's make it a bit more interesting in the next one. Let's say we had information for the point types. So as I was mentioning in the previous, well, in the workflow one, let's say we had information of blood hunts, and we know that mosquitoes, female mosquitoes at least, would be more likely to be shifting from aquatic habitat to a blood hunt. So we encode that information into this additional matrix, which is the probability of the female mosquito to jump from aquatic to aquatic. In this case, it would be 20%. It would all quite obviously be more likely to fly to a blood hunt, and vice versa, to jump from blood hunt to aquatic and to blood hunt to blood hunt. This is obviously not calibrated. I just made the numbers up for the demonstration, but let's say we had those data, and we can include that into embedded in our landscape, and this is how it would look. The framework automatically would assign a different icon for every point type, and the links between the nodes would shift just because the probabilities vary. So even though these 2 points are closer to each other, it's way more likely for a mosquito to fly in between this, and this one is almost non-existent because they wouldn't be flying between those as likely just because of the way we define this one. And the time to capture all adapts accordingly. So in this case, it would probably be better if this trap were here, this one here, and this one here, or some combination of that. But we're not optimizing it right now, so it's just a demonstration at this point. I don't know if there's any questions or comments so far. Anything has come up?
Hector, there's a question from Matthew in the chat.
Oh, yep. I can't see.
Can you hear me now?
Yeah.
Okay, yeah, the question is about whether this is optimizing trapping any mosquitoes, or is it optimizing catching the gene drive individuals, and would that be able to change over time as the gene drive spreads?
Yeah, at least if no changes are made, the way it's been set up right now, this would optimize catching any mosquitoes. So the idea would be that you lay the traps and it would try and catch any mosquitoes in the hopes to detect the gene drive mosquitoes. But if we have some evidence that gene drive mosquitoes fly differently, let's say, I don't know, they fly shorter distances, we can incorporate that information to optimize either wild mosquitoes, gene-drive mosquitoes, or both at the same time. I don't have an example in this one because that's a bit more contrived, but the way that can be set up is optimizing two landscapes at the same time. In the documentation, I have an example on optimizing for male and female mosquitoes, which would be sort of similar in this case. So if we, for example, female mosquitoes do go to blood hunts in aquatic habitats, males, simplifying a lot, let's say they don't, we can optimize the two landscapes at the same time. Just mix them together with a combination of the fitness functions. And we can also do that. We haven't done it because it's a bit more complicated, but we have examples of that running in our documentation, and that could also be done for a gene drive versus wild ones if that's one of the applications that could come up.
Great, thank you.
Yep, no worries. And yeah, so the final one in terms of landscape is the one that I was mentioning with a more realistic one, because right now most of the ones I've shown are toy examples of just flat surfaces. And this one is the one that we will be using the migration matrix that Thomas Leon generated for some years back. So for example, if we have the latitude and longitude coordinates of the landscape, and we want the landscape to be plotted with the geographical features and whatnot, we can easily do it. We just use another library, Cartopy, and we can plot it with the boundaries and whatnot. And in this case, for example, one thing that's different from the other ones is that we are getting the points from a CSV file, a static CSV file anywhere, but this migration matrix, the links between all the points, are not being calculated by MGSurvE. These were calculated by Thomas's algorithm. There's a link to that one. I think I did it here. Otherwise, I put it here to his website and his poster on that one. Oh yeah, there's more information on all the notebooks if anyone's interested. So yeah, that migration matrix is being fed to the algorithm, and that can be done easily. And we're doing the same thing. In this case, we're optimizing 10 traps. All of the traps are set to the same type. And we are optimizing this one discretely. So traps can only be placed in sites that are defined in the landscape. Otherwise, we just can't. It's not possible for the algorithm. This usually runs a bit faster. I'm running it for 25 generations, which is extremely low. It's usually like 1,000, but this is a larger landscape, so it would take way more time to run. But here we can see that even with 25 generations, the algorithm is like placing traps in reasonable places. Here there's a disconnected set of two disconnected land masses on top, north and south. So the algorithm identifies that there should be traps there because otherwise we wouldn't minimize time. And it's spreading them as much as possible. This one, it usually places a bunch here because there's a lot of really connected points here. I think it's not that visible in this plot just because it's small, but there's a lot of connected points there. So it usually places a lot there just to catch as many as possible. But this is one of the landscapes we are using actually in our paper. We in our all of our preprint examples are also available here. So if anyone wants to look at them and even run them, it's right here. So both the Jerky's Nova example and the Sao Tome one, which I just showed. This is another one that does a similar thing, but with different traps. So we have the landscape of households, and we're doing the optimization of the traps. We can see that it tends to spread them. Again, this one is just run 250 generations, so it's not quite long enough. It's usually like 2,000 for this one, but it's doing a good job in spreading out the traps. Just close this one. And that's pretty much it for the landscape. I don't know if there's any questions on that. Otherwise, I'll be going to a bit more in detail to the optimization and of the pipeline. This one's a bit more dense, but yeah, I don't know if there's any questions on the first one. And if there's any questions in the chat, could someone tell me? I just can't find where the chat is in my. Oh yeah, here it is. I think there's none. Okay. Oh. Otherwise, let's keep going. So in terms of the optimization, that's a more complicated end of the pipeline. and rightfully so. It's the meat of the dish, I guess. But the way MGSurvE works is it defines a set of population of, well, of synthetic population of possible solutions in which each individual contains the information of the trap's locations. So, for example, each chromosome, each possible solution would contain the information of each trap location. Let's say it was 10 traps. It would have the 10 traps located somewhere in the landscape. And if it detected that, well, it would usually perform 3 operations on this. It would be the let me check if I have them in order. Yeah, actually it is here. Initially, it would calculate the fitness of each possible solution. So given that the traps are located somehow in the landscape, it would calculate the fitness, the time to capture, which would be this one. Then, after it does that, it selects stochastically from the population. It would assign more probability to possible solutions with better fitness, with better solutions to get picked for the next iteration of the cycle. So in that way, it would guide the optimization to converge to a certain optimum solution. And once it selected this subset of the population, it would do the crossover operation. So it would mix the genotypes of the possible solutions between the coordinates of the trap. This is different for the discrete and continuous one, but in general, it tries to mix the two parents genotypes into a possible solution of locations of traps. And then it does some mutation algorithm, which adds some randomness to the solution so that it can explore the landscape. And that's it. After doing this over 1,000 generations or so, it will hopefully converge to a good solution. How good the solution is depends on the landscape and a lot of factors of how probable, well, how much we increase the mutation rate, the crossover rate, the selection rate, and that usually takes a lot of a bit of fiddling around with parameters and a lot of just general knowledge on running these things a million times and figuring out the best parameters. But in MGSurvE, we have set up some parameters that are good in general for a rule of thumb parameters, either from literature or from our own experience for the discrete and continuous cases. So if we don't want to set them up ourselves, the mutation rate, crossover, and whatnot, we can just set up the algorithm to run in auto mode. I think one of these should be in auto mode, if I'm not recalling correctly. And no, I did set it for that one. The quick start should be auto mode. Yeah, so we can just set them up to auto. It would do its best, and we can start tweaking them later. But the other parameters usually work reasonably well. I think those are the ones we use in the paper, but I'm not entirely sure. But yeah, for example, I'm not gonna run this one either because it takes a couple of it takes like 1 minute, but in any case. But in this case, we will be running a grid of points. Another feature of MGSurvE is that we do provide the tools to set up synthetic landscapes for grids. The donut one, uniform landscape, random uniform, and Poisson, and I think another one, but I can't remember. So if you want to benchmark your traps to see that it's working, you can easily do it. But in this case, we set up 2 types of traps in a grid, and we can see that even just after 300 generations, it's optimizing them pretty well. Like this is a benchmarking algorithm in the discrete one. This is what we would usually expect from a grid, so we can check that it's actually running correctly. And we can do the same for the continuous one. In this case, I added a bit of spice to it and had it with different point types. So there's 2 point types, and it shifted a bit the solution. Well, it shifted the solution a bit. But yeah, same thing. I think I also run it for, I ran it for 1,000. So yeah, in this case, once the landscape is set up, we can just launch our optimization algorithm with these couple of commands, and that's it. We just wait for a couple of minutes for it to finish, and we're good. It will usually plot our landscape, and we can also inspect the logbook, the generations and whatnot, and we will do so for one of the analysis notebooks. I don't know if there's any questions on this optimization, and I know this one's a bit more dense just because it's the complicated part of this.
Hey Hector, I just have a question. So I'm just curious, like, are there any heuristics on like whether your algorithm's not converging or if it's like stuck in some local minima? Because I'm curious from like my work in machine learning, like if your stochastic gradient descent just stops working, like it'll just run forever, but like the actual outputs will be really bad. So I'm curious, like, how do you assess that sort of thing?
Yeah, let me check. I have it. Well, I have a couple here. Yeah, in general, it can get stuck. It depends on the landscape. For example, in the uniform ones, it's really rare that it gets stuck. It's just it usually keeps improving up to the point where it's just like, okay, this is good enough. Within the optimization framework, there's a couple of ways you can go about it. One of them is that it does have the option. This would need a couple of extensions, but it's not difficult. One of them would be to have it stop at some point if it's not getting enough improvement, like the same as you usually do with stochastic gradient descent. If it's not improving a lot, you just stop it and rerun. Right now, the way we're going about it is more, let's just run it for 1,000 generations or 2,000 and run a bunch of repetitions, like, let's say, I think in this one is at least 10 for each one, but it can be 20 or whatever, and check against each other. There's another one, which one of the undergrads from Mexico implemented with particle swarm optimization, which was and I think it's in the code, I just haven't had a chance to use it, adding simulated annealing to the genetic algorithm. So beginning in the first phases of the algorithm, the mutation and crossover rates are high, but as it keeps going through generations, the mutation and crossover rates start decreasing so that it's more stable. And that has the advantage that first you can explore a lot more, and then in the end it just converges to a more stable solution. I think that's in the codes somewhere, or otherwise I would just need to look it up. But that's another way to go about it. But yeah,
that makes sense, because I think in like machine learning, if you start with like a high learning rate and then you decay it over time. So I think it's similar to that. Okay, cool.
Yeah, exactly. Yeah. That's the probably the best. It's a bit more complicated. It uses the particle swarm one instead of using the genetic algorithm. We have implemented this group of undergrads implemented the particle swarms version of it. It runs fairly well. Oh, this example is really small, but that one I think does include the simulated annealing. So yeah. I don't know if there are any other questions on the optimization end of things or anything else.
I have maybe it's beyond the scope of this talk, but hi Hector, by the way. Thanks for your support. So you've shown how to optimize the placement of traps, but I'm still missing the step of how do you come to this landscape? Like, how do you find the landscape to inform your placements of traps? Because it feels
Oh,
like
yeah.
you need the traps to go to what is the actual landscape.
Yeah, in this case, it depends a bit, but for example, let me check one of the more realistic ones. The Yerkes Knob one. In this case, the way we're getting these sites are the houses. So we got these placements of the sites from parsing the OpenStreetMap data. Let me show one of the demos. So for example, actually, this is one of the other things I've been working on lately, parsing information from OpenStreetMaps and using this as the sites. So for example, in this case, we're parsing, I think this is Burkina Faso. I just don't remember the place exactly. But we can use those information, those data, to use it as the sites. In the case of the Sao Tome, for example, this one, Thomas did the work. He, if I don't recall correctly, and he grabbed the actual map from the location, and he clustered the villages in some way that was reasonable for the mosquito movement, human movement, and the census data. And that's how we ended up with these ones. It depends a bit. So in these two cases, that was the case. I think there's another one in which was. Yeah, so for example, this is another one that I'm working on in OpenStreetMap, but doing some more clustering because it's too large to run itself. But yeah, this would be part of it. But in these examples, we are assuming the resources are available to mosquitoes somewhat uniformly. So those data should probably be gathered on the field because otherwise it's just, yeah.
Oh, wait, and when you say those data, you mean geographic? Or what data?
Yeah, so for example, one thing that at least in my small work with OpenStreetMap, it's usually not specific enough to be like, oh, this is a fountain, this is, I don't know, it's usually, this is a building, this is a building. Some regions have better data, but it's not like, oh, this is aquatic, this is nothing, it doesn't have enough information to be like, oh, I can assume this is aquatic, this is it. So we have just done the uniform thing up to this point, but if that data was available, it could be incorporated.
And so, sorry, I don't want to hijack. No, but say you do a mark release capture. Is there any plan in the roadmap to use that to infirm the landscape? That yeah,
I know.
With traps, given traps, using that information to actually optimize the or figure out how the mosquitoes travel and how connected areas are.
Yeah, I see where that was. Yeah, I think I went about the question the other way. Yeah, I can see that one. If you were planning a mark release recapture, depending on the shape of the landscape, well, I don't know. It also depends a bit on the methods to do the migration on the mark release recapture, because I'm not that acquainted with how you get the population sizes and the migration from the MRR. Yeah, but we could probably figure out a way to do that for a trial. In this case, it would be more like just trying to minimize the time, not so much to optimize so that you get the best results for the migration distances. So yeah, I can see the disconnect between those two, but we can probably talk about it and figure out a way to incorporate it to make it a bit more useful for MRRs, for example.
Okay.
Yeah, because this was initially more oriented towards detecting transgenes as quickly as possible, but we can probably talk about it and figure out a way to make it useful. So yeah.
Great. Thank you very much for that.
Yeah, no worries. Any other questions? Otherwise, we can go to the final part. This one's a bit more lightweight. Finally, after we optimize our landscapes, we usually want to visualize how the traps were located and how the algorithm performed. Do some maps, even videos. I don't think I have videos here, but maybe here. Yeah, so for example, we can export even videos for how the algorithm is behaving over time, how it is trying to optimize the location. And it's all included in the framework. The videos one is a bit more complicated because it takes time, but all the other stuff is right here. We can plot our attraction kernels for the traps, the migration kernels, as I showed in a couple of slides, the landscapes. We can plot all of them with the IDs of the nodes, the locations, the fitness. We can even plot the maps, as I was showing previously, with the OpenStreetMap data included. We're still working a bit more on the OpenStreetMap end of the spectrum because, as we can see, these landscapes become really large. So there's some work to do in clustering and aggregating the landscapes so that they can be optimized in larger setting. Otherwise, it starts becoming computationally feasible to do. So we have to come up with a couple of tricks on that end, but we're looking forward to working on that. And finally, we usually compare the as I was mentioning with Agustin, we compare the behavior of the algorithms in some shape, well, similar to this. In this case, we are comparing how the time to detection gets lower as we add traps. So for example, this one is just 5 traps, and it would be the purple one here. But if we do, I think it's 5, 10, 15, and 20, yeah. We can see how it's getting optimized and how the traps are getting more dense over the whole landscape, and how this time to detection starts going down. But we can also see that at some point the difference between 15 and 20 is probably not that much. So we can start thinking of costs at that point. Like, is it really necessary to add 5 more traps just to get this slight benefit? Because as we add more and more traps, the benefit starts. Well, the return for investment is probably at some point not worth it. So we're also starting to think in terms of that and thinking about how to incorporate it directly into the optimization algorithm. That's also another interesting area of research in this framework. But yeah, usually we benchmark the algorithm's performance with these plots. It's the number of generations we run the algorithm through, and the fitness. We generally want the fitness to jump around in the first stages because that means the algorithm is exploring, but to become stable after a while and stop jumping around at the end, which is what we're seeing. It's just generally good to have. But we can also have a look at the logbook, at the evolution of the whole thing. If we have some suspicion that it's getting stuck up, as Agustia was mentioning, or that something's going wrong, we can check out all the logging. Everything gets exported to disk, and whatnot can be checked by anyone later on. And I think that's it in terms of the main stages. I added more information in more notebooks on the repo, just in case anyone was curious to get a bit more deep into, let's say, the math or some of the how it incorporates OpenStreetMap's data, the preprint or whatnot. But that would mainly be it on my end. I don't know if there's any questions or comments about the MGSurvE framework. I know I went through it a bit quick on it, but well, it's a lot of both. So yeah, but yeah, I'll give some time for questions or comments.
Can I ask another question if no one has a question?
for sure.
So you guys seem to be putting a lot of work into making this accessible. It's quite a niche field. So do you also have ideas of making this like targeting regulators and decision makers, giving them the tools to like independently assess these type of projects?
We could try. Well, in that regard, it could be done. So, for example, as you mentioned, this is still a bit niche. Like you do need to go a bit into the code, even though we're trying to make it as accessible as possible. The code still needs to get done. Like there's no way around it right now. This could be extended somewhat to be a bit more standalone, like to provide an interface for someone to provide a user interface on top of some basic examples, or with the OpenStreetMap thing, to couple it to do some optimization just with a couple of clicks here and there. One of the limiting factors right now is that it is computationally intensive. Like I didn't show it here, but for example, one of the reviewers a couple of reviewers actually asked for the runtime of a benchmark of the runtime of the optimization, and it looks sort of like this in terms of number of traps and number of sites. So it does take like a couple of hours to do a realistic landscape for one repetition. So we would still need to do a bit more work on optimizing that, or to put it out on a server that can run that automatically, or we would need to give it a bit more thought and work towards making it more accessible, let's say with the user interface or something like that. But it could be done. It's just thinking about that one.
Cool.
Sure. Thanks. And yes, Fredros, you have a question?
Yes, I have actually two questions. The second one will depend on whether the first one is satisfactory. So my first question is actually a request for you to simplify this whole thing and just say what the objective of the modeling framework is, and what you wish to see in the end. So I want to ask that as a question, which is, are you building a mechanism to guide the placement of, or the identification of surveillance sites and the placement of traps?
Yeah. So, for example, one of the applications, I'm going to use the Sao Tome one as an example.
Right. Yeah.
So, for example, one of the things we would be interested in this case would be, let's say we are planning on a field release
Correct.
trial in this case, and we would be releasing, let's say, on the south. Our first question here is like, let's say we have 10 traps and we want to know where to place them to detect it, to be able to track the spread of the gene drive as it sweeps across the island.
Correct.
We would want to be able to track it as uniformly as possible. We can't just be like, oh, we could do just a grid and place here to her, but given that we know mosquitoes don't fly equally to every place, we can do that. So this is the first step in that pipeline. But yeah, I forgot to mention this before. This only tells us where to place them, generally speaking, if mosquitoes were behaving according to some simplifications.
Correct.
But the second step in this case would be, and Agas is going to talk about it in a bit more in a second, is to export these migration matrices and feed them into a more realistic, into a model that has the mosquito life history, the mating, and all the mosquito components, like MGDrive in this case, which Agastia will mention in a second. That way, we can also simulate and know how much actual time it would take and how often we would expect mosquitoes to fall into traps and at what times we would expect to be catching them with.
But essentially, at the beginning, you want to use this to identify the most appropriate places to put your 10 traps, correct?
Aha, exactly. Yeah,
Okay.
yeah.
Okay.
Yeah. Yeah, I think.
So the second question then is in terms of adaptability. So how does the result of your first placement influence the second placement? So if you have initially you have 10 traps and your matrix and all the other covariates, based on that you decide that you're going to place them in places X, Y, Z. Depending on the findings of the first traps, you want to adjust the placement of the subsequent 10, correct?
Sorry, can you repeat how the first and the second relate to each other?
So in the beginning, my understanding was that in the beginning you have a clean plate and you want to figure out, okay, where should I put my first 10 traps?
Uh-huh.
So does the result of those first 10 traps influence the placement of the subsequent 10 traps?
Oh, if we wanted to add traps, or if we wanted to
if
shift
you wanted
them.
to, because you're going to expand these surveillance systems, right? As the gene drive spreads, you want to put more traps in different places, right?
Yeah. If, for example, well, it depends a bit. So if, for example, this trial had 10 traps at the beginning, then we're releasing in between, we add 10 traps on top of this. You could add them. One of the things this framework can do in that case is mark the first 10 traps as fixed. I didn't mention it. I kind of forgot. But for example, these dark crosses mean that those traps can't be moved. So if you have locations that you're like, okay, these 10 traps have to be here, and I have 10 more traps that I want to add on top of this, you can optimize the second 10 with the first 10 just being immovable. And then the second 10 would be the new batch to optimize around it. So that could be done that way.
I'm satisfied. So that's, if you allow me just one last quick question on that.
Yeah.
Yes. So I guess the point I was raising here is that in the beginning you do not know the direction to which the constructs, the direction that the constructs will take. You are making that assumption based on certain knowledge of the landscape and mosquito behavior. However, when you put your first set of traps, they will give you some data that could inform. So the point I was raising is, could you make this a little bit more adaptable so that the findings of that first set of trapping can inform the subsequent expansion of your trapping matrix, right?
Yeah. So if that was the case, it would be more like, okay, I placed them here, then I have new information on how mosquitoes are flying. So you could just take the landscape, grab those data, calculate the new migration matrix. Let's say we put the traps in 10 locations somewhere. Then after a month, we figure that mosquitoes are actually not flying there. They are flying in a different pattern. Then if we gather those data and can translate it into some migration matrix, we can get rid of this one, then feed this into the algorithm and optimize the new traps locations with the new data and try it out again, let's say a month after that, and check if that holds true and it does catch more, we can stick with that, or we can keep iterating. As long as there's a way to calculate how mosquitoes are moving within the landscape, that can just be, let's say, an iterative process within the framework. Yep, no problem. Yeah, I haven't thought about that, but yeah, that's a neat.
Yeah, I think that also directly goes to my question of like, if you do a mark release recapture with these traps, and then use that information to build the landscape.
Yeah, exactly. Another thing that, for example, could be done before doing the MRR would be to run the simulation with MGDrive or another simulation that has a life history, and use that to feed into MGSurvE to optimize traps location, then do the releases, and then do it again, iterating as we were mentioning. That could be another way to go about it. But yeah. That's. I'm gonna keep thinking about that one because that's an interesting. Don't know if there's any other questions or comments. Otherwise, I think we can go on with Agastia's end of the presentation. How do I stop sharing? That's it.
Cool. Thanks, Hector. Yeah, hey everyone. Thanks for joining. I'll be talking a little bit about MGDrivEv3, which is kind of our linked entomological epidemiological framework for simulating genetic vector control interventions. Let me share some slides I made. Let's see. Okay. Can you guys see my slides?
Yep.
Cool. Yeah, so like Hector mentioned, this is kind of our complement package to MGSurvE, more focused on estimating the epidemiological impacts of genetic vector control interventions. So yeah, just to go through what I'll be discussing here. And yeah, I'm not able to see the chat, so if you have any questions, just call them out. Yeah, so I'll just be talking a little bit about the prior work and what has kind of led up to this framework, and talk about the overview of the software architecture and some of the new changes we've made since the last version was released a couple of years ago. I'll walk through a couple example simulations, some very basic ones, just to show like kind of how you would go about setting up one of these kind of simulations, with the caveat that there are a lot more aspects to the software that I'm not covering here, but are available in either the CRAN page, or I'm happy to chat offline if there's a specific use case that you would like to use this framework for. We could work through that together. And then, yeah, just talk through some future direction and what we're thinking about in terms of next steps, and just leave some time for questions. So previously, our group developed MGDrive to model the spatial population dynamics of a variety of genetic vector control systems, and MGDrivEv3, which kind of linked those population dynamics to simplified epidemiological dynamics and seasonality. Here we present MGDrivEv3, which takes kind of the core components of those previous software packages. It has three kind of major components that it adds. The first is a new sampling algorithm that allows the vector and human components of the model to be formulated in whatever way that they need to be formulated, and can be readily modularized and swapped out for different models if needed. And I'll talk through how we did that. And we linked the entomological model with a version of the Imperial College model of malaria transmission, which is a more complex model than the one that was included in MGDrivEv3, which incorporates age structure, immunity, different human and vector interventions, and more meaningful disease outcomes, and a surveillance module that tracks the number of mosquitoes caught by traps across a geospatial landscape. And yeah, in this session, I'll just describe some of the architecture and how to set up some simulations. And like I said, more documentation can be found on CRAN. So the first aspect of this is the decoupled sampling that I mentioned. So generally in vector models, you have a vector component which tracks the life history of the vector through egg, larva, pupal, and adult stages. And then you have a human component which tracks the different infection states, whether susceptible, infected, recovered, whatever it may be. So these models just pass back and forth the relevant information to each other as the simulation progresses, but each one is kind of agnostic to how the other one is formulated. So this gives us kind of the benefit of retaining the way that MGDrivEv3 developed a stochastic entomological model for the life history, which is kind of a discrete state space Markov model using something called a stochastic Petri net, which is like a computationally efficient way to store Markov models. And we are able to link it with a continuous space partial differential equation model for the epidemiological and human infection dynamics. So in this iteration, we paired the model from MGDrivEv3 with the Imperial College malaria model, and we made some modifications to allow the gene drive aspect of it to be simulated. And here's just a schematic of how that kind of works. So, each model is separate, and then they pass back and forth the forces of infection at each time step. And yeah, like I mentioned, because they are only dependent on that one parameter, the force of infection, you could, in theory, swap out the vector model for, maybe a different vector whose gene, if you want to model a gene drive intervention for, or if you're interested in something like arbovirus transmission for dengue or for Aedes aegypti, dengue, yellow fever, those kinds of disease transmission models. In theory, you'd be able to do that in this framework without too much work. So just to talk briefly about the different models we're using here. So the gene inheritance and vector life history models were developed previously in MGDrive and MGDrivEv3. Like I said, they utilize the discrete state space Markov system to stochastically model the movement between different life history stages, stratified by genotype. So here the users can specify which gene drive construct to model, whether it's a CRISPR-based homing drive, something like a suppression drive using sterile insect technique. You can write your own inheritance pattern. So it's all very flexible to what your specific use case can be. And yeah, the epidemiological model is adapted from the Imperial College model, initially published in 2010. Schematic here is below. And it just tracks infection through various states from susceptible to treated or untreated disease, protected, asymptomatic, and subpatent infection. And there's various rates and parameters that go into that. But there's just kind of a highly simplified version of the schematic there. So how we modified this to kind of incorporate the gene drive aspect. The entomological component of MGDrive is genotype stratified. So we just include a genotype specific force of infection that gets applied to the human model. So here's just the math from the paper, but essentially if you have a set of genotypes, capital G, and then the proportion at a given time in the time step, we just calculate the total transmission probability to be the weighted average, and then that gets fed into the force of infection. So in the example we'll show, which is a kind of CRISPR-based homing replacement drive, all the mosquitoes in the simulation after the drive is released are conferred with a gene that prevents the transmission of disease. So then the overall force of infection is decreased, and then that subsequently affects the prevalence and incidence of malaria in the model. And so the previous model just tracked prevalence and incidence using like a simple kind of SCIR framework. But here we updated the model to be age structured, so we can calculate outcomes for different age groups, and especially the 2 to 10 year age group, which is often reported in malaria studies because pediatric cases of malaria tend to be the most severe. We track prevalence using the number of humans that are in a compartment that is still infected. So whether it's a treated or untreated asymptomatic disease, asymptomatic but undetectable, or asymptomatic by detectable disease, we're able to keep track of all of these by age. So that's one of the big improvements from the last iteration of the model. Sorry, I see a question in the chat. Oh, sorry, that's cool. Thanks. Cool. And so another major component of this framework is adding seasonality and baseline intervention. So generally, when we're designing a simulation for gene drive intervention, we want to accurately capture the location in which the trial might take place. So there are a lot of aspects to that. One is that seasonal, weather patterns are going to affect how mosquito life history evolves over time. So in the previous version of MGDrivEv3, we used daily rainfall data to model how larval carrying capacity would be affected by rainfall, and that worked. But often it's kind of computationally demanding to get a lot of rainfall data, and it can be very noisy depending on the location. So in this version, we used a library called Umbrella, which was also developed by Imperial College. And what it does is it fits a mixture of sinusoids to the daily rainfall data. So it allows you to capture the dynamics more broadly in what they're calling kind of a seasonality profile. And we use that to calculate the larval carrying capacity instead of the daily data. And I think there are cases where daily data might actually be preferable, maybe if you're modeling like a very specific field trial location with really small geospatial resolution. And this framework is still flexible enough to do that, but we thought for kind of these initial use cases to not have to download years of data, this kind of mixture of sinusoid approach would be preferable. And then another aspect of characterizing the location in which a genetic intervention may take place is, these will be deployed alongside existing vector control interventions, such as insecticide-treated bed nets, indoor residual spraying, and some disease management interventions as well, such as the probability of receiving antimalarial treatment upon infection. So now we can specify all of these different things, and they will appropriately modify the different parameters in the life history and the epidemiological model. One aspect of this, which we're thinking about expanding, is that these are kind of static coverages, but as we know, with things like resistance and just products not degrading over time, it would be useful to have time varying efficacies of these, where for maybe even just like an exponential decay or something like that. But right now they're just static, and they modify all of the relevant parameters before the gene drive intervention would take place. And yeah, finally, the last aspect that MGDrivEv3 incorporates is a spatial modeling component, which allows the placement of traps across a geospatial network. As Hector mentioned, as Hector discussed in his MGSurvE package, we have optimized kind of this MGDrivEv3 framework to accept input from MGSurvE. Right now, it's a little crude in that you still have to do some pre-processing to make it work, but we're trying to streamline that a little bit. But ideally, you would decide where your traps wanted to be placed using MGSurvE's optimization engine. You could specify the type of trap, whether it's an odor-baited trap or a light trap or whatever, and then you can parameterize the different trapping probabilities all in MGSurvE, and then export the optimized placement of traps, and then feed that into MGDrive to really simulate the entire surveillance program. And we provide different functions to simulate emptying traps in different timeframes, whether you want to check your traps weekly or monthly or whatever it is. This framework allows for that flexibility. So, overall, I think it's just in conjunction with MGSurvE, we want to be able to inform the design of an intervention and surveillance program for gene drive products. So I'll just quickly go through a couple of simulations here. So the first one is a single node epidemiological dynamic. We'll walk through how you would set up a homing-based population replacement gene drive construct in Anopheles colutzii. This represents the TP13 replacement construct that was recently published by Tony James's lab. It's a CRISPR-based homing gene drive linked to dual disease refractory genes. So any mosquito carrying the transgene has a reduced probability of transmitting the malaria pathogen. And we will just demonstrate how you'd set up a basic release of that and in a single node. So it's not geospatially accurate, but you can capture just the high-level dynamics of how you'd expect this to behave. And we will show how you'd parametrize this to the seasonality and baseline interventions of the island of Sao Tome. And all the code for this is available on GitHub, so I wouldn't focus too much on the code, just kind of more so the workflow overall. If there's any questions, let me know. Otherwise, I'll get into the code here. Okay, great. So the first part of this is just a bit of setup here. So and this is R, just to clarify. So yeah, Hector's package was using Python, but this is mostly in R. And we are kind of thinking about different ways to incorporate faster programming languages like C and C++ into this workflow, but for now it's all R. So yeah, we first just load the libraries and then we source the cube. So the cube is what determines how the gene drive spreads across generations. So this cube was specifically developed for this construct. Rodrigo, who's a postdoc in our lab, wrote out all of the different probabilities, but essentially what it does is just tracks the probability of different offspring getting the gene drive across generations. But MGDrive actually gives us a lot of cubes out of the box. So if you have a simple construct, you don't need to write your own. You can just take a look at the MGDrive documentation, and they have cubes for homing-based drives, for suppression techniques, and other ones. So and then we load in some external data. The E matrix is just the genotype-specific transmission blocking parameters. So those were also fitted to lab data when we were building out the simulations for this construct. But essentially all it says is that if you are a wild-type mosquito, you have X probability of transmitting the pathogen and given a bite. If you have the transgene, this is the probability. If you have a resistant allele, et cetera. So yeah, again, you don't need to write your own, but if you have a custom construct that you are fitting to data, then that's typically how it'd be done. And then just some basic simulation parameters, the total simulation length, the stochastic and deterministic time steps. The stochastic time step is a little finicky in that if it's very small, then the simulation results are very numerically accurate to a high degree, but it takes a much longer time to run the simulation. But if it's too large, then you can get some wonky results, especially if you have small values of disease prevalence to begin with. So it's kind of you have to play around with that a bit. I think we're looking into how to kind of optimize that as well. And then, yeah, the next step is kind of setting up the baseline parameters for the simulation. So the cube, we instantiate the TP13 cube with a 10% fitness cost. So that's that FC parameter. And then the probabilities are the ones that are fitted to data. not super important, but if you look at the documentation for whatever cube you're using, then you can see what parameters are required to instantiate that cube. And then we define some baseline parameters of the location. So the prevalence of malaria, which is 2%, which we've just pulled from Malaria Atlas project. And then we're just doing a simulation in 1,000 humans. You can set that to be a accurate population, but generally I think it's easier to just set it to be 1,000 and then scale it as needed later on. And then the imperial model param list create function just has like all of the parameters required for the imperial model to set up its baseline parameters. You can modify the individual parameters as needed, so they have like life history parameters, different immunity profiles, and if you look at the documentation for that function, you can just see all the different parameters, and if you need to modify any of them for a specific strain or species or anything, you can do that there. Then the age vector is just the distribution of age groups that you want to consider. So here is just between 0 and 5, 5 and 17, 17 and 40, 60, whatever. And then that the 1,000 humans will get distributed amongst those age groups in a like an exponential way. And then finally, the vector control and disease management interventions pre-GeneDrive, so the IRS, LLIN, and anti-malarial drug coverages can be specified there. And then the add interventions function just modifies the relevant parameters based on those. So yeah, that's how you'd set up kind of the location in which you want the simulation to take place. And then the next step is determining which determining the Markov states of the model. So these functions are just provided to you. You don't really need to go too in depth into this, but if you pass in your inheritance cube and your parameters, it'll set up all of the different states in the Markov model using the SPNP and SPNT functions. And then using that, you can get the equilibrium values for all of the different states using the age vector and the different parameters and the baseline EIR. So this isn't super important. If you're interested in the math behind this, I would take a look at the supplementary information in the MGDrivEv3 paper, which talks about how the different states are set up and how the movement between them happens. But yeah, for the simulation, this is just something that sets all of that up. And then the hazard rates are the transition rates between the different states. So I wasn't able to put the whole code here because it's kind of long, but I would take a look at the full example. But essentially what we do is we read in the seasonality profile from, we read in the seasonality profile from the rainfall data, which is calculated separately. And then we make time-varying hazard rates for larval carrying capacity based on that. So the transition function for larval carrying capacity is time-varying dependent on the seasonality. And then we just use the make hazard function to create all the different transition rates between different states. So in this case, only the larval carrying capacity one is time varying and the rest are constant, but you can write your own to make any of them time varying dependent on external data based on your use case. And there are examples of that in the CRAN and GitHub. So just going over it from a high level here, but yeah, I would encourage you to take a look at those examples later on if you are curious on how to write custom hazard functions. And then finally, we just define the release strategy of the GeneDrive. So in this scheme, we're releasing 20,000 homozygote male mosquitoes every week for 8 releases starting at year 10 in the simulation. And you just need to create this events data frame, which just has basically the type of release you want, and then at what time in the simulation you want. And yeah, that's pretty much it. So then once you run the simulation, here's the function to run it. There are a couple of different functions. So this is the one that just runs in R locally. But generally for large scale simulations, if you're doing simulations over a bunch of values, there's a function that writes to CSV files. So generally we'll use that one to write all the data to CSV files and then analyze it separately using either Python, or you could use R to analyze the data. It doesn't really matter. I won't go through all of the parameters there, but essentially you just pass it in everything we set up, and it'll run the simulation. If you want to do stochastic repetitions there's a way to do that using the CSV function where you set up as many different stochastic repetitions as you want, and then there's a couple additional functions that will calculate the mean and the quantiles of those stochastic simulations. And yeah, the human ODE parameter here determines the human infection dynamics. So here we're using imperial. There's also the SEIR, I believe, if you would want a simplified version of that, and SIS model as well, if you want an even simpler version of that. So yeah, that's the kind of overview of the epidemiological dynamics. Here's the figure we created for our paper. So this data we analyzed using the Monet DA package that Hector developed. It's just another complementary package that allows you to analyze large streams of data coming out of MGDrive. So if you have, if you're trying a bunch of different parameters and things like that, I would encourage you to look at that package to better wrangle all of the data. But yeah, as you can see in this scenario, we have the gene drive system spreading rapidly following the release, and pretty much the entire population being replaced with the H allele, and a small proportion of resistant broken alleles. We calculate the age stratified clinical incidence per 100,000, and as well as the prevalence. And we see their decline after the initiation of the gene drive system. Yeah, and of course these are highly simplified. They don't take into account the different spatial dynamics or anything like that. Though that this is more to just be informative on the parameter sets that would lead to a potential elimination scenario, or the time scale depending on the immunity profile or the baseline prevalence of a given area. The prevalence we used here was a very low starting prevalence, which is why we kind of see this rapid elimination. But yeah, it's not meant to be a kind of like an extremely accurate depiction of what may happen. Of course, there's other species and environmental drivers, but it's just more to inform some of the higher level parameters of what a gene drive intervention could look like in a given location. So yeah, that was super fast. Obviously, there are a lot of different aspects to this, and once again, happy to chat offline if there's a specific use case that I can help out with. But that's a high level how you would go about setting up one of these epidemiological simulations. We are thinking about different ways to incorporate spatial dynamics into this. It's just, as Hector mentioned, these are pretty computationally intensive, and I think once you add a spatial dimension, it can get even more computationally intensive. So we need kind of smarter ways to think about that. But the part of the model where we do consider spatial dynamics is the trapping, as Hector discussed in the last segment. So I'll just kind of walk through quickly how we would set up a spatial simulation using traps, optimized trap landscape based on the routines in MGSurvE, and how we would feed that into MGDrive and what the output looks like here. So here I'm just using a very simple inheritance cube, not a custom one at all. So if you see this, the first line of this code, that cube is provided by MGDrive directly, so you don't even need to write your own. And then some baseline parameters of the entomological model, the death rates and the dwell times in each aquatic stage. And then some simulation parameters, how long you want to run the simulation, the stochastic, or the time step. And then we read in this CSV that was exported from MGSurvE, which gives us the coordinates of all of the different points. We're using the same landscape of Sao Tome, and just for the sake of computational efficiency, we're just filtering to the south of Sao Tome, using the latitude and longitude values there. We specify which index is the node you want to release release the intervention in, and then the trap nodes, the type is -1, so we just keep track of which are trap nodes and which are population nodes. And then this raw migration matrix gives us the daily pairwise probability of movement between nodes. So I'm just reading that in and just formatting it to make it nice. So this part we are planning on streamlining, so you don't have to actually do this yourself. We'll just have functions that let you read in whatever is exported from MGSurvE, and it'll just feed it in and do all this behind the scenes. But we didn't have time to set that up before this, so I'm just showing you right now how it would be done. So then we construct an adjacency matrix from the movement probabilities. Essentially, if there is a non-zero probability of movement between two nodes, then we say they are adjacent, and that just helps us set up all of the different Markov states and lets us define which movements are actually possible in the model. So then again, we pass it into the SPN functions and calculate the different equilibrium states. And we set the number of female adult mosquitoes in each node. So this is just arbitrary right now. So the actual results aren't accurate to any real scenario. But I think ideally, if you had data from your trap experiments or previous mosquito density data, you could feed that in here to more kind of accurately capture the baseline distribution of mosquitoes in each population center. And then, so this is another thing that you don't have to worry about too much that we're going to move to behind the scenes, but essentially we have to renormalize the matrix to convert it from a discrete time probability space to a continuous time rate space. So we just calculate the rates based on the probabilities and then renormalize the matrix. But that's just so that the Markov model can use a continuous time approximation instead of the discrete one. But yeah, if you're interested in the math, again, I would take a look at the MGDrivEv3 supplementary material. But yeah, this all will be behind the scenes going forward, so not too important there. And then again, we determine the hazard functions. Right now, we're not doing any time-varying hazards, so these are all just the default static hazards. But if any of them, again, you want to be externally driven by data, then you can parameterize that there and write your own custom hazard functions for that. And then again, we set up the events vector or matrix, which just says the release time, the release scheme. The only thing that's a little different here is that we have to specify which node we're releasing in. So that's the 7th node corresponding to a population center in the south of the island. And then, yeah, this function will run it. Again, we have versions that will do this in CSV files. So these spatial simulations do tend to take longer, especially if you're running a bunch of different nodes. It can take a couple hours or even overnight. So I would encourage to run CSV files so you can just have them whenever it finishes and analyze the data from there. And yeah, here's the example of that from the paper. So we just simulated traps along the southern part of the island. Again, this is not optimizing any of the placement of the traps. Those are exported directly from MGSurvE. So these are just static traps. And then the trap count is kind of arbitrary. Like I said, it's not actually parameterized to any real situation, which is why it seems quite high. But just for the sake of demonstration. And yeah, as you can see, all of the traps were able to capture a transgene, but the timing and the amount of mosquitoes are a function of the distance from the release node. We didn't mark the release node here, but it's the very most southern blue dot on the island. And yeah, that's pretty much it. I would say if you are setting up one of these simulations, again, to use the CSV functionality and take a look at the Monet DA package, which lets you do kind of this visualization in a nicer way than fiddling around with R. And yeah, just quickly to talk about some future directions. So we're exploring the use of the simulation software for the development of target product profiles for gene-drive interventions, kind of modeling a variety of different scenarios in terms of different baseline intensity, transmission intensities, different genetic parameters such as homing rates and the resistant allele generation, and trying to quantify their impact on these different epidemiological outcomes of interest. Like I mentioned, we're also interested in improving the computational efficiency of this framework via lower level programming languages and emulation techniques. I'm working on a project with some folks at the Gates Foundation, trying to think through if you can build emulators for these really complicated models that are based in machine learning, then it might be faster than running the full set of models themselves. And then, yeah, converting some of these functions to C and C++. There are R packages that allow you to interface with those lower level languages, can speed up the computation. So that would be highly beneficial, especially if you are considering running large suites of simulations. And yeah, overall, we just hope that researchers will adapt this framework to other models of vector-borne disease transmission. Like I mentioned, both the human and entomological models are able to be readily modularized. I mean, and this is all open source, so things like ADs and arboviruses, or agricultural pests, and just genetic interventions for vector control more broadly. We hope that there's an interest in adapting this framework for that. And yeah, that's about it from me. I think, yeah, I went super fast just to kind of give a high level of how you would go about setting something up like this. But yeah, happy to take questions and chat offline if there's any more. Yeah, if there's any specific use case I can help out with. And if not, I'll pass it back to John.
Okay, well, thanks everyone. Thanks, Gus. I guess you can wait a couple more minutes for questions. But yeah, thank you so much, Hector and Augusta, for the overviews of the MGSurvE and the V3 packages. And yeah, those are really great presentations. Hopefully people found it useful. And as Augusta mentioned, particularly for the more involved simulations like the ones he presented and Hector presented, it might be easier to follow up in person afterwards on that. Yeah, but we're pretty close to 10, which is our planned time. Maybe, yeah, any final questions from anyone?
I have one. So you guys are building very advanced models. I think the most advanced in the field. do you guys have a philosophy at what point you get ahead of like the biological information and where the biologists start to fail you and we just can't provide the needed information and the needed input for these models?
I think it's often the case with models that they are overspecified. As in there's more detail in the models than than has been provided by the ecologist or epidemiologist at this point. And so I guess that guides our approach to try and develop relatively parsimonious models. But there has also been a call to incorporate, more ecological, in particular, detail in the models. And to do that, it will vary a lot by field site, and then we'll want, like you're mentioning with the mark release recapture, having that data to inform the spatial models will be really valuable. I guess we aim to keep up with the data that's becoming available. And there would be places where there's a little more, but modeling generally would be an iterative process as well, as Hector pointed out earlier. So whenever additional data does become available, then I think it's really important that we have dialogue with the people collecting that data and making sure that it's reflected in our models. I don't know if you want to add anything, Hector and Augustia.
No, I think, well, that goes along the lines that I was going to mention. I guess we try and build it. Well, at least in my end, I usually try and think of well, I guess we all do, the model, making it as flexible as possible. On my end, it's usually because of the goals. Like there's usually decisions that need to be made, like, we can do this, but if this kind of data becomes available in the future, we won't be able to use it. So we better try and make it flexible in this case, just in case we do have the data. But yeah, for example, in the case of MGSurvE, the point types and all of that, it's there, but even, for example, reviewers have asked why we didn't use resources in our examples, and I just don't know how to parameterize it. Like it's there, the capability is there, but it's a bit tricky. So yeah, I guess we try and make it as flexible as possible in case the data becomes available, but sometimes it does, sometimes it doesn't, but it's there. So yeah.
Are there any aspects you've been able to validate so far? Like obviously not with an actual homing gene drive, but with any other type of studies?
I think the closest one would be, well, the one we did for the first version of MGDrive with the Wolbachia releases in Yorkie Snuff. We did some benchmarking with that so that we could at least get a similar response to that. And I think that would be the closest one in terms of spatial experiments, at least the releases, at least. But yeah, I think that.
Yeah, on the lab side or on the construct side, then that's where we have the most ability to validate our models, just because it's a relatively simple system. And we have time series data from all the gene drive intergenerational experiments where we can fit the models and validate the parameters. Those are parameters in the lab rather than in the field, but they are validated against something at least, and we'd iterate on that in terms of the fitness, which we would expect to differ in the field. And another point I'd make on the epidemiology is that one of the reasons we've adopted a malaria model from another group is that they've validated their model to data. The imperial model has been validated to data all throughout sub-Saharan Africa. There's still a lot of assumptions and a lot of parameters in the model which are just taken as given, but it has been validated against available data sets for disease endemic countries in the continent. So yeah, so there is data that's been validated against, but there is still a lot of room for improvement as that becomes available.
And I tend to hijack these things, but are there things that experimentally you've already identified that, oh, if you can measure these while you're doing your preliminary scoping of an area or your first field trial without an autonomous drive, are there certain unexpected parameters that we can capture?
I mean, I think as John mentioned, the fitness cost of these gene drive systems are pretty not well understood, especially in the field. Like for the construct that we modeled, the TP13 construct, we had some estimates of fitness from the lab, but they didn't seem super realistic, or we just weren't confident that that's how it would behave in the field. So we just simulated a couple different hypothetical values. But I think, yeah, once a field trial is done and we have better measurements of that, that would really help to inform the models, because we found that the epidemiological outcomes are pretty sensitive to the fitness of the gene drives. So that's, from my perspective, at least one thing that I think these models can get much better once we have better data on that.
Yeah, and ecology, of course, is far more complex than we can understand at the moment. So there'll be some details there which would be great to understand better. Yeah, likely whether there are cryptic mosquito populations where the drive may not reach or something, or yeah, the movement patterns like Hector was mentioning. There are data that, yeah, I guess mark release recapture will be a great proxy where that's available.
Yeah, I wonder if these things that it might not even be that useful to measure them, because you can't measure everything and there's always going to be experimental errors. So I think it would be useful if these models, you could instead give them the answer and they sort of back compute because the easiest thing we might be able to measure is the spread of the gene drive in these traps, and all the ecological aspects we might not. So maybe we want to estimate parameters on a small scale and then use the model to predict them on a larger scale. Fedros has a question.
Yeah. Can you hear me?
Yes.
Yeah, a minor question here. You probably already addressed it earlier. But I'm wondering about the actual component of the package that you're interested in following here, whether it's the GeneDrive construct itself or just the Cas9, especially in cases where, let's say, you have a GeneDrive resistance. You still want to monitor the spread anyway and figure this out. So is there been any considerations as to what aspect of the technology you want to monitor here? And I have a different question, which is from the manuscript that you guys shared. In the manuscript, you talk a lot about safety, but I couldn't figure out what's the safety question there really. I thought it was just monitoring is good nonetheless. It's not necessarily just for purposes of safety, but the way it's presented, it's almost like the technology is risky and therefore there are all these safety concerns, but there are no explicit safety questions raised in the manuscript. So just those two questions.
So which was the first question again?
So first question would how do you incorporate for the loss of the construct?
Or the loss of the construct.
So yeah.
And you might have a lingering Cas allele, for instance.
Correct. Yeah. So is this optimized to monitor the Cas9 or to monitor the GeneDrive construct itself?
Yeah. That would yeah, so for modeling that is relatively straightforward in the MGDrive framework, just because the different genotypes can be modeled, and so you can have genotypes corresponding to the Cas9 with or without the guide RNAs and the effector alleles. So we do have the flexibility to model just the Cas9 or the guide RNAs or both or neither. And for the safety, then the surveillance is motivated by both the efficiency of a field trial or intervention and how best to monitor that, and also to monitor potentially the emergence of, say, resistance alleles or the spread of gene-drive alleles to places where you don't want them to spread, say, beyond the field site. Both of those questions can be, both of those categories of questions can be addressed using the surveillance that Hector and Augusta have described. So yeah, you're right. We don't want to be expressing this as overemphasizing the safety issue, but probably emphasizing both the safety alongside the efficiency.
Thanks a lot. Last question. So is it always going to be the Imperial College model embedded to this, or this could be if you wanted to say open malaria, for example, could this work with that, or it's always going to be the default with the Imperial College model on top of your system?
I mean, right now it's that, but I think it's, if someone wanted to go in and link it with open malaria, that'd be doable. Like I mentioned, the models are pretty separate. They only require a couple of parameters, so it's definitely doable to link it with open malaria. I don't know if that's in the plan right now, but yeah.
Thank you, guys. Have a wonderful day.
Thank you.
Thanks, Vedras. Yeah, thanks so much, Augusta and Hector. And thanks for the questions, Sebald.
Thank you very much for the presentation.
Great. Okay, well, have a great rest of the day, everyone.
Bye. Thank you for coming.
Take care. Thanks. Bye-bye.